Thank you. So, friends, please allow me to introduce Anil Kumar, who is an associate professor at Vignan University in Guntur. He has been my good friend over the last 15, 16 years. He is a PhD in plant biotechnology from Osmania University Department of Genetics in 2015, where he worked with Professor Emeritus Kavikishore sir, uh, where Kavikishore sir is well known as doyen of plant genetics. Anil worked on various plant-related genomics in terms of sorghum and as well as sodium potassium portals towards the abiotic stress traits. He has also worked on uh, other animal genomics and viral genomic related entities, taking cognizance of plant specific uh, uh, phytochemicals. He has also been associated as Bioclues research fellow, as a visiting fellow, which we call them as AP Bionic Overseas Research Advancement Program to Singapore, where he and Ima were there for 4 months as a part of their PhD program. Later on, he had his postdoctoral stint, and he has been associated with Vignan over the last 6 years as an assistant professor and now professor. He is also a core member of Bioclues, serving as an outreach chair. So today, he is going to introduce us to the field of plant bioinformatics, where he'll give a talk, and then that is followed by hands-on session until 8:30 p.m. Thank you so much.
Yeah, thank you, Dr. Prashant, for the introduction. Hope I'm audible, right?
Yeah,
Yes,
yes,
sir.
Anil.
To everyone. Okay, before going into the topic, first I would like to say, all of you please attend the workshop from your laptop rather than your mobiles or some tabs like thing. Tab is okay, but better to join through laptop because straight away we'll move on to hands-on, and in between wherever it is necessary, I will explain the content. I'll explain the content, later we'll move to hands-on session. Again, I'll explain some more content. Again, you need to practice. It's completely 85% based on exercise and 15% based on theory. Okay? Is this okay?
So, excuse me, sir, I think your voice is echoing and it seems like you are speaking
Oh, okay.
from some
Please
distance.
give me a minute.
No, it was fine for me, sir.
Wait, I'll use earphones instead. Give me a minute. Is this okay now?
Yes, sir.
Yeah, fine.
Yes, sir. It's okay.
Yeah, good evening to all the participants. A warm welcome to you for this workshop. Straight away, we'll move on to the topic, not wasting much time on introduction and all those things. Already, President has given a brief introduction about me. In today's class, I will explain you about one of my research areas, that is genome-wide analysis. Going to the paper, coming to the slide. This is a paper which I published in the year last year around September. The paper
Sir,
is about
sir,
hello.
please share your screen.
Okay, already shared. I will reshare. Entire screen. Okay. Can you see my screen?
Yes, sir, it's visible.
Yes, now we
Okay,
can see.
fine. Already I had in the chat box I had shared the article also. You can open the article in your respective systems. Here is the link for that particular paper. I had opened that one. Please open that one simultaneously can check. I will explain, but keep it with you. Yeah, coming to the presentation, this is the paper which I am going to explain. I mean, in today's hands-on, purely it's 85%. Already I told you it's 85% hands-on, and only 15% will be on the lecture. I'm going to explain on this particular paper. It's about genome-wide. I mean, in this paper, we had identified how many genes of that particular family are there in sorghum plant. And even we tested whether the genes are responding to particular abiotic stress or not. So the crux of this paper would be we are trying to mine the genes from a genome, and later we are going to verify and validate whether they are really the same genes or not with the in silico tools, followed by their expression patterns or the transcript profiling, or simply doing QRT PCR will validate them. Now coming to this particular paper, I will show the full length paper at once. This is the full length paper. Can you see the paper?
And the film paper is not visible. Anil.
Hello.
Your voice is breaking badly, Anil.
Guys, are you able to see the paper?
Yeah, open it, please. Your voice is breaking.
I'm audible.
Yes, sir, you're audible. We could see the desktop. No, ma'am, it was breaking for me as well.
Yes, sir.
Yes, your voice is very clear, ma'am.
Not mine, Anil sir's.
Yes. Otherwise, I think whatever the headphone or something they have used earlier, it was okay
Yeah.
for Anil sir.
I mean, I could see his screen, but his voice was breaking a lot. Sir?
Yeah, good evening, Ankita. Yeah.
Sir, I'm not able to join from laptop, so can I join by phone?
Yeah,
I
yeah,
think some
Ankita.
issues are there.
Yeah, it's okay, Ankita, but the problem is, Anil is going to give you a hands-on, since it's a workshop, over the last several days. So it's okay, but you can do those exercises maybe after whenever you use a laptop. Okay. Yeah.
Yeah, sure, sir. Actually, I'm trying, but some technical issues are there.
Right, right. Yeah, sure. It's okay.
Yeah, thank you, sir. Yeah.
When 2 weeks ago, Otter AI chatbots have ruined the entire organization's emails. You know, you can just Google it. That's how the serious offenses. Even they are not to be trustworthy at all. Okay.
So I have one question. So we have to include the topics of till 29th, or we have to
Yeah, until
close it
28th.
from today.
29th is an extension of what Manjari has suggested. So what Manjari spoke yesterday. So it's a meta review part 2. So you can cover until tomorrow. Okay. Even aaj ka bhi chalega, but you have to make a good story. Okay. Whatever.
Okay, one more thing, sir. In Jairam sir lecture, one of the student of Jairam sir also given one presentation,
Right. Right.
but we didn't receive that presentation. So could you
Okay.
please send it?
Sure. Sure. Okay. Yeah, I sent a message. Huh?
Okay, sir. Yes.
Yeah, I'm sorry for this delay. I think Anil has had some serious connection problems. Huh?
So those 5 slides will be containing the results of our exercises or
Yeah.
what exactly?
So, for example, you have done some exercises and you can make a story, Pranita. Okay. So you can probably make, so maybe you might have queried something. Okay. You've gotten some results. So you can correct 2 or 3 other exercises as well. Okay.
Okay, okay.
That thing also you can do that. Yeah. Thank you everyone, for taking this workshop very seriously. Thank you so much. I appreciate that.
Sir, actually, we should thank you. You're teaching us like even many universities may not be teaching this much.
Yeah, thank you.
Uh,
It's all the efforts of all the faculty here. That's how our workshops have been actually. We take these attendance and everything very seriously, and
that's why we are also learning seriously, sir.
and that is probably the reason why we were a bit harsh on those members who joined without paying 2,000. So there's no free thing anywhere. So even if someone gives anything for free, that is not warranted. So, so that is how we take these things very seriously. Even the NGS workshops or even the previous workshops, even in fact the integrated systems bioinformatics 2021 workshop was a huge hit. There, we had about 40 members, and all the 40 members gave an excellent presentations. We had to take presentations for 2 days, and we had to thank all the speakers and for their time and energy. And it will be a good this thing. The pity is my own PhD fellows they didn't attend this actually. That is a very sad story. Yeah. Hello, Anil. Yeah, Anil joined through Hima's account.
Can you hear me?
Yeah, yes, Anil. Yeah.
I'm really sorry. My system got completely stuck off and it's not switching on. So I joined through other system. Yeah, straight away moving to the article. Please, in the charts box already I shared the article. It's about genome-wide analysis of potassium transporters. Please open that article. All of you, please open that article. I'm sharing my screen. Yeah. Can you see my screen?
Yes, yes, Anil.
Yeah, thank you, and I sincerely apologize for this delay. Now, if you see the paper, in this paper, we identified how many total potassium transporters are there in that particular gene family and in sorghum bicolor. So, by end of this hands-on, I wish whoever is interested in this kind of research, choose your own family of genes so that we can work on genome-wide analysis. The take-home message from this paper would be we selected a group of genes, belonging to a particular family, that is potassium transport, this potassium transport, and we had identified homologs in sorghum. Before starting this work, we had taken the gene sequences from the published paper, that is genome-wide analysis in Oryza sativa, nothing but your rice plant. Around 49 genes are there in that one. One of my seniors carried out that work, and later I extended that work in sorghum. All you need to do is you need to take the potassium transporter gene list, the available list, and you can start mining the data in sorghum. For that, I think all of you are logged in through your laptops only, right?
Yes.
I'm audible to you.
Yes. We've logged in through laptop.
Okay. First, we'll go with the gene of our interest, and later you can work on gene of your interest, because first I will show how to take one gene, and then I will explain you how to mine the data, how to check whether that what are the data that you have mined is of the same gene or not. Okay, is this clear? First thing is, first we'll take a template sequence, then we'll go with the mining of that sequence in the plan you wish to. Okay, and thirdly we'll validate that one. Okay, all of you open NCBA. Who are lagging behind, please let me know in the chat box so that I'll stop there itself. Please go with the same gene for the test run, and later you can choose your gene of interest. Okay, open NCBI. Once you open NCBI, hope you are able to see my screen, right?
Yes, sir, it's visible.
Yeah, fine.
Yes,
Yes, sir.
Yeah, thank you. And I'm typing a gene by name NHX1. NHX stands for sodium proton antiporter 1. And now I will check this one in Arabidopsis. Or suppose you have gene from Arabidopsis, and if you want to work in sorghum, I will show you how you can work it on. Yeah. You can take partial, but better go with the complete sequence. Or we'll take the first one. Take the faster sequence. Arrows maybe in third or fourth page. Hope all of you are in the same page, right? Are you able to understand? First, we are taking NCBI, open NCBI. Once you open NCBI, collect gene sequence, a gene sequence which you want to study. And then open Grameen database.
Sir, Grameen will be having only one family.
Are you able to
plant.
follow me? If not, please let me know. I will stop there for some time so that everyone can move together.
Sir, one doubt, sir.
I am audible to you?
Yes, sir. Yes, sir.
Am I audible to you?
Are we audible, Anil, for you?
Sir, I
Hello?
think you couldn't hear us.
Okay, thank you. Thank you, Dheeraj and Rajat. Thank you. Fine.
Sir, can you hear us?
Once you take the faster sequence, open
Sir?
the Grameen database.
Sir,
Why
I
we are
think
opening
you couldn't.
Grameen database? Grameen database is a platform for almost all plant genomes. What are the plant genomes that have been sequenced? They will be there in the Grameen database.
Sir,
Now
can you
this
hear us? Sir, sir, can you hear us?
from Starmella.
Uh,
I
to
want
raise
to
a hand and once he sees,
identify
then
or
you
discover
speak.
its homologue in sorghum.
Yasmin.
I
Perfect. What I
joined
will do
in
is
mobile, madam, so I couldn't do that.
and
Can you
open
No,
please?
no,
Grameen
he's not able to
database.
listen to us. That's
Then
so
click
clear.
on blast. Click on blast. Then submit your sequence. Submit your sequence. I'll off my camera so that Pranita Legume Lab. May I know who is the Legume Lab?
Anil,
Yeah, Manjari.
you're not able to hear us.
I can hear you.
You just now you're able to, but earlier you couldn't.
Sir, actually, this is Yasin.
Yes, madam. Yeah, please.
Actually, Grameen will be having only one family crops. It is not for all plants, no, sir. So, can we use some other database?
Earlier when started, they used to, archive only Poaceae family.
Okay.
The grass family.
Okay.
Right? Now
Yeah.
they are adding with FTP other additional sites. You can get those things also. I'll show you in the.
Okay. Okay, sir.
Yeah, fine. Now moving back to the Grameen database, we had taken a sequence and we are looking for homologue or isoform of the homologue of that particular sequence in another species. For that, you open Grameen database. Okay. In Grameen database, open the blast. After opening the blast, you submit your sequence here. You can name the sequence SNHX1, something like this, or SBNHX1. Okay, fine. After this, by default, here it is chosen as Arabidopsis. Please change that one. Now I want to identify in sorghum plant or rice plant. What are the plant you are interested in? You can choose that one. But for the test one, all of you choose the same, whatever I choose, so that we'll get the same result. Later on, you can exercise your gene of interest. Type here sorghum bicolor. Okay. Once you get sorghum bicolor, check the box. After checking that box, again check here, so that your species will come here. Earlier it was Arabidopsis. Now you can see sorghum bicolor. Is this clear? At a time, you can submit up to 30 sequences. You can submit up to 30 sequences. For demo purpose, I'm showing only one sequence. Is this clear?
Okay, sir.
Is this clear?
Okay, sir.
Yes, Anil, yes.
Yeah, good, Peter, you have raised your hand. Please let me know your doubt.
It's a mistake. It's clear. Thank you. I've lowered my hand.
Can you repeat the question?
No, it was a mistake. It seems, sir. You can please continue.
Okay, fine. All of you are in the same page with me?
Yes, sir, we are. But
Yes, sir.
the gene which is selected by you is exactly what we have selected.
Okay, fine.
Because we have
Because
selected one mRNA from I think from Arabidopsis for NHX1.
okay, you can select any gene, that's not an issue.
Oh,
But the problem is, after characterizing the gene, I may get 3 exons, you may get 7 exons, then there may be some discrepancy. What I am
okay.
suggesting is, if everyone, all of us will go with the same gene, then it would be easy in analyzing the result. That's my intention. You can select any gene of your interest, there's no issue. But analyzing the result, I keep on saying 3 exons, 2 introns, something like that. You may say, I have some different thing. That is the problem.
Okay.
Or else I will copy and paste the same sequence in the chat box. Please use the same sequence. It would be better.
Yeah.
Yeah.
Because I have selected already, but I don't know which
No,
one.
no. I shared the sequence in the chat box. Please use that one. Right? Copy and paste the same sequence in the box, in the sequence data box. Then select
I think
sorghum
you've
bicolor.
missed the first line. It is not in FASTA format. You missed the first line?
In the first line, it has contained the
It's not
accession input and
important.
other things are there. So I had
Okay,
kept
okay.
only the sequence.
Fine.
Yeah. Okay. Once you submit, choose your genome of interest as sorghum for this exercise. Then we are using DNA sequence. No need. Then we'll go with the genomic sequence and nucleotide blast. We can go with the same search, whatever that are there in the default parameters. Just click run. Once you click the run, it will take around 30 to 40 seconds and you will get the result. You can see here the status here. It's queued, so it's running. It will get automatically refresh for every 5 to 10 seconds. And once the result is ready, it will display the result. Done. Now the result has come. Hope all of you also got the same result, right? Yes or no?
It's running, sir.
It's running fine. I'll wait.
Got, sir. Similar. 6 hits.
Okay. Did everyone get the same result? Or is anybody still doing the same thing? Those who are completed, please wait for your friends. Hope everyone is in the same page, right? You got 6 results. Just click on the view results. Okay. Once you click on the view results, here you can see the genome location. From the genome location itself, we can say whatever we are searching for that homolog, it is located on chromosome 6. Sorghum will have 10 chromosomes, and whatever that you are searching now, it is located on chromosome number 6, and it is located in between this position. From here to here, it is located. Is this clear? Yes, sir. Everyone, if someone is lagging, please let me know so that we can wait till you complete.
So
Yes,
my
sir.
net is slow. My net is not working properly.
Okay,
So I
we'll
think
wait for. Uh, I can take class up to 8 to 8:15. Don't worry about the time. If you do not have any other meetings, I want to show how to mine, how to validate whether you have mined the same homolog, I mean the gene homolog you're looking for or not, with the help of some domain proteins and other things. Right?
yeah, okay.
Once you're done, please let me know. Because if you keep on moving further and further and someone is lagging behind, then it will be difficult for them to understand what we are doing. Yeah, once you're done, please let me know so that I will
Yeah,
move
yeah,
to the
sure. Yeah, sure.
page.
So I got it. Now we have to go to view results.
Yeah. Once you
Yes.
click on view results, the page
Yes,
will
sir.
be displayed like this, whatever exactly the same whatever I'm showing on the screen. Okay. And from this result itself, we can say here it is 100% identical. Okay. And mostly 99% or even 100% also sometimes, our hit will be, I mean, the best hit will be on the top position. Best hit will be on the top position. From this, you can say that whatever the gene homolog that you are looking for in sorghum is located on chromosome number 6, right? And it is located in between this particular location. Now simply click on the sequence. Click on the sequence so that you will get some sequence, right? Is this clear? Is this clear to everyone?
Yes, sir.
Everyone?
Yes, sir.
Okay. Now, what is the actual length of this particular sequence? Go back here. You can see it is 1623 bases. We have taken 1623 bases of sequence by a gene by name NHX1. We had blasted it against sorghum bicolor, and we landed in a particular chromosome. And this is the position of that particular chromosome. Now you configure this page because this particular sequence is very small. You configure this page. Once you click configure page, you can see the parameters. Number of bases per row is 60. Default, it will take around 300 bases upstream and 300 bases downstream of your blast hit portion. Now make it to 3000. Because of my experience, I am saying 3000. Once you practice with 2 or 3 sequences, you'll also come to know why we need to take at least 3000. Okay. At the same time, move down. There is an option by name line numbering. Just keep it as none because we cannot keep on deleting the line numbers in each and every line. Once you choose none in the line number, check this again so that it will get refreshed on its own, and now you will get a
Just a minute. Just a minute. What did you do after you opened the sequence in Grameen?
open the sequence in
Yeah.
Grameen. Okay.
No, after view results, you click the
Okay.
first sequence.
Yeah,
Then
we clicked on the first sequence. the first sequence is almost 8 to 10 lines, right?
Yes.
Yes. By default, it will take around only 300 bases upstream and 300 bases downstream of the core portion. So we are increasing the length of this one by configuring the page. On the left hand side, you can see configure this page. You please click that one. Are you able to get? I will show you again. Check this one, configure this page. Okay. And now make the 300 to 3000 and 3000. Upstream to the blast hit portion, we are taking 3000 bases, and downstream we are taking another 3000 bases. Is this okay?
Yes.
Yeah. And if you see the previous sequence, there are line numberings are there. In the line numbering option, click none. Because we don't want line numbers. If the line numbers keeps on appearing, it would eat away our time to remove the line numbers itself. So we are keeping it as none. Is this clear? Once you click none, check this check mark or the tick mark. Now you are getting a much bigger sequence than your earlier sequence, right? Guys, is this clear?
We got this.
If you have any doubts, please ask me. We'll clear here itself. Is this clear to everyone?
Sir,
Yeah, please.
after setting this
Yes, sir.
line number, no, what you did?
In the line numbering, choose none.
None. Okay, that we have done.
Okay, on the top side, you will get the tick mark, right, of that particular page.
Okay.
Please check that one. Tick that one again, so that it will get refreshed. And now we are going to get a much bigger sequence without line numbers. Right? Yes. Say something, or else I don't understand whether you're following me or not. Please let me know whether you're following or not. Yes, Rakesh. Yes, Dheeraj.
Yes, sir.
Okay,
Yes, sir.
people. Done. Hassan?
Yes.
How about Hassan? And
Yes,
10
sir.
more people are there. Okay, Hassan, yes. Yeah, you, Madam Pranita?
Yes, yes.
Yeah, fine. Then, now I'll show you. Now we got a much bigger sequence. This sequence contains your exons, your introns, your promoter sequence, your terminator sequence, whatnot. We don't know what are the entire sequences that are there. I mean, what are the portions that are there in this particular sequence. You simply copy this one, and in the next tab, open GenScan. GENSCAN, GenScan, which will scan your gene. Hence, it is GenScan. Open this particular link, new GenScan web server at MIT. Okay. As we are working with plant systems, choose Arabidopsis. You can even choose maize also, but Arabidopsis is more annotated when compared to maize, and more duplications are there in maize. Better and submit here. No need of any faster format, just submit here. You'll get the result like this. You'll get the result. Okay, you'll get the result like this. By looking at the result, we can come to some conclusions. See here, the length here 1623 base pairs. Now here it is around 1200. So, and at the same time, you look clearly at this particular CDS sequence. A CDS can be called as CDS only if it satisfies three conditions. The first one is it should start with ATG, your start codon, and it should end with any one of the stop codon like TAA, TAG, and TGA. Is this clear?
Sir, please, pardon, repeat. Can you repeat, sir?
Yeah, I will do. See, listen, or else you can see this sequence, whatever that has been submitted in the GenBank. See here. Here it is written as NHX1 complete CDS, coding sequence. For a sequence to be called as coding sequence, it should satisfy three criteria. The first one is it should start with start codon, that is ATG. I'll increase the screen size. Okay. It should start with ATG. It should stop with any one of the three stop codons like TAA, TAG, or TGA. Okay. Here you can see it is starting with ATG. It is stopping with TGA. And the whole value, whatever particular length, it should be divisible by 3 because 3 nucleotides code for 1 amino acid. Is this clear?
Yes, sir.
Okay. Now coming back to our own result, whatever we have done through Grameen database. We had submitted that one to GenScan. We got around 1200 base pairs. Now it is starting with ATG. Fine. The start codon is there, but there is no stop codon, right? Can you see here?
Yes, sir.
Your start codon is here, ATG, but there is no stop codon. Here you can see. Either TAA or TAG or TGA should be there. Instead of those, it is ending with GAG. What does it mean? We had taken a sequence where we have not taken the 3' end. For that, what I will do is again go back to the blast page. Okay, go back to the blast page. Now configure this page again. In the previous, we had taken 3000. Now I'll go with the 4000. You can straight away choose 5000 also. Straight away we'll go to 5000. We'll check whether it will work or not. Okay, and then check this box again. Now you'll get much more bigger sequence than the previous sequence. I am copying that particular sequence, and again I am pasting the same thing in the GenScan. Run the GenScan and check your result. Guys, can you see the result?
Yes, sir, it's visible.
Now the thing here is, when initially we had taken upstream 3000 bases and downstream 3000 bases, we got a start codon, but we did not get a stop codon. For any gene or any functional CDS or any CDS to be called CDS, it should have start, stop, and divisible by 3. Now in this sequence, you can see it is having a start codon, right? It is also having a stop codon. Is this clear?
Yes,
Yes, sir.
but why we didn't do this in the first step only?
Generally, most of the time, if the gene is around 1500 base pairs or 1200 base pairs, if you take 3000 upstream and 3000 downstream, most of the times you'll get the complete CDS. For this gene, the introns may be bigger. We don't know. Now we need to check how bigger the introns are there. That's why we are configuring the page again so that we can get 5000 upstream and 5000 downstream to get the complete CDS. Initially also you can take 5000 at a time, but sometimes what happens is if the genes are very close to each other, some other gene and this NHX1 gene, we will ran into the previous gene or the next gene. So it's always advisable to better start with the lower configuration and move to the higher till you achieve your complete CDS. Is this clear?
Correct. Correct. Yes. Yes. Correct.
Yeah,
Thank you.
fine. Now we have a sequence now. I mean the complete CDS now. Keep it aside. I am saying that this is NHX gene. You may get it out. What is the proof that why we should call it NHX? In order to call it as NHX, this particular CDS or this protein should contain domains which helps in sequestering sodium or simply reducing the sodium levels in the plant system. These days, whenever we water the plants, salt goes inside the plant. As sodium is toxic, even for human beings, also the same thing is applicable. All the time, we need high potassium levels and low sodium levels in the body. Okay, if the sodium increases, obviously it leads to blood pressure in human beings. The same thing happens in the plant system also, not blood, but their metabolic activity will be disturbed. So the plants try to send the sodium away from the plant, either into the soil or it will send it into the vacuole. On the whole, the plant tries to keep the cytoplasm of the cell in electroneutrality condition. Means irrespective of the water conditions available outside, plants try to maintain the pH of the cytoplasm so that the normal metabolic functions will occur without any disturbance. Now, first I will show you what is the domain that is there in this particular gene, for which you open next tab and go with CDD search, or you can take smart. We will go with the CDD search, conserved DNA domain search. Okay, please open this one, CDD search. Once you open CDDA search, you can see here, click this one, or you can open this one also, anything is okay. Why I am explaining this is, first we'll verify what type of the domain that is there in the template sequence we have taken, and we'll validate our sequence, whether what are the sequence that we have retrieved or mined from sorghum bicolor is showing the same domain or not. Because all the time we need to validate, because there are only ATCG, 4 bases are there. It's just repetition of all the genomes with the only 4 bases. Sometimes there may be a possibility that we may get a similar sequence, but it do not belong to the same family. So you copy the sequence, the template sequence, whatever you had taken for blasting against Grameen database, copy this one and paste that one in CDD search. You can submit. I think you need to submit in faster format. Submit the sequence and checks its function. Check its function.
So can you just share this again? What you have told? I just lost the connection, so I missed.
Yeah, fine. I will repeat. No issue. I will repeat. Now the question is, we had taken a template sequence. I told you to take NHX1. You had taken that one and you had blasted that one against sorghum bicolor plant. You got some sequence and you have with the help of GenScan, again you got the CDS sequence. This is now what we have done up to now, right? Yes.
Yes, sir. Yes, sir.
Yeah. Now we need to check whether whatever the sequence that we got is the right one or not. We need to validate that one straight away. Okay, based on the GenScan output or based on this particular sequence, you cannot say, okay, well, I got the sequence. You may got the right sequence, but it need to be validated before going to the next step. For that, what I'm saying is, first take the template sequence, whatever you have taken, and submit it in the CDD search, conserved domain search, right? Once you submit into that the database, see here, this is the result. It is showing sodium hydrogen exchanger 3. Can you see this one? This is with the template sequence. Can you
Yes,
see
sir.
this one?
Yes, sir.
All of you, please respond
Yes,
because
sir.
I'm not understanding whether you're following or not.
Yeah, sir. Yes.
Everyone, if anyone has any problem, please let me know so that I'll stop there and we will practice that one again. And now, okay, this is having some domain or description like sodium hydrogen exchanger. Now, what I will do is I will take my sequence. Okay, I mean my sequence means whatever we have retrieved now with GenScan, right? Copy this sequence and paste the same sequence in the CDD search box and submit.
So you have copied the sequence from GenScan.
Sorry, I have not copied the complete sequence. Copy the complete sequence.
I didn't get this, sir. I means in the GenScan, the last codon is not the stop codon in my case.
Uh, you should get the stop codon. Have you taken 5,000 upstream and 5,000 downstream?
4,000. I've taken 4,000 upstream and 5,000
That's
downstream.
what. Please go with the 5,000 and 5,000 because it may be there at 4003 position. Right? Hassan, go back to that configure page. Take 5,000 upstream and 5,000 downstream. Okay? And then paste that sequence in GenScan. You will get the CDS sequence. Once you get the CDS sequence, submit that CDS sequence in
Sir, once can you please show that in GenScan again, please?
GenScan.
Yes, sir.
Okay. I'll go back again. What are the sequence that you got in Grameen database? Okay, as a blast output result, copy that one. Go to GenScan. Choose the plant Arabidopsis and take both CDS and peptides. Submit your sequence here. Run the GenScan. Right?
Okay, sir.
Then you will get the result like this. Once you get the result, you can see it is having the start codon ATG here. Okay. As well as it is having the stop codon TGA at the other end. Now we can call it as a CDS. What I am doing now is I am checking whether what are the result that has been given by GenScan is the right or not. How do you check that one? We need to check for the presence of domain in that particular sequence. Presence of domain in that particular sequence. I'm just copying the sequence and I'm pasting it in CDD search. You can even use Smart, SMART, Smart. There is another tool by name Smart domain. This is a smart page. You can even use this also. CDD search with both proteins and nucleotide sequence. On the other hand, Smart works with only protein sequence. Is this clear? I'm submitting the protein
Clear.
Smart.
Sir,
Yeah, please.
4,000 upstream, 5,000 upstream, वो समझ में नहीं आया, sir. That's why I asked you to show.
Pardon? Can you repeat your question?
No, you explained something to Ziaul. 4,000 upstream.
Yeah, no. While configuring the page, I asked you to take 5,000 upstream and 5,000 downstream. When Hassan has taken 4,000 upstream and 4,000 downstream, he did not get that one. Okay, the stop codon. So I am saying you take 4,000 upstream and 4,000 downstream.
Sir, वो कहाँ से करना है, sir?
Again, configuring the same page. Initially, I have taken 3000 upstream and 3000 downstream, right? Okay, I'll show here. See here. Again, I'm coming back to blast genome sequence. Configure this page. Here, take 5000 upstream and 5000 downstream.
Okay, sir. Okay, sir. Okay, sir.
Okay.
Okay, sir. Thanks, sir.
Then you will get much bigger sequence than your earlier sequence. Earlier, you had taken 3000 upstream and 3000 downstream. Now you are going with 5000 upstream and 5000 downstream. Check that box. The page will get automatically refreshed on its own, right?
Okay, sir.
And then
And this is the reason why this referred to Grameen instead of directly in NCB blast.
yes.
Okay, sir.
Okay.
So can you show the CDD search again, because I'm not getting any result in this. Yeah, only this one is coming. Search result successfully.
Okay. Open CDD search. Okay. Conserved domains database. In that one, click this one, CDD search. The advantage with CDD search is you can use either protein or nucleotide sequence. There are some other tools like PFAM Smart where you need to use only protein, right? Before going into that particular thing, we need to check whether what are the sequence that you have copied, what are the sequence that you have used as template for discovering a homolog in sorghum. What feature does it exhibit? Simply submit your sequence here. Remember, you need to submit in FASTA format. Submit your
So you
sequence.
have copied the sequence from NCBI or from GenScan?
NCBA, because we need to know what type of that function that particular CDS is displaying, right? And we need to check this whether the same function is there in GenScan output or not. Are you able to understand the concept?
Yes, yes.
Listen, I will explain again. This is a sequence we have taken, right, as template from GenScan. Sorry, from NCBI. We had taken this one and we had blasted against sorghum in Grameen database where we got some blast output result. We had taken this result and we had submitted that one to GenScan so that you got some gene sequence like this, right? 2052. Yes.
Yes, sir.
Fine. Now it's only repetition of 4 bases ATCG, ATCG all through the genome. We need to verify when you're identifying new genes whether that new gene has the same function of the previous gene or not. Gene with the help of CDD search. Submit your sequence in CDD search and go for this button submit. Once this button is once the result is submitted, you can see can you see here the function sodium hydrogen exchanger? Everyone, this is for your template sequence, right?
So what result you got for smart? I did not do smart.
Smart, I know I did not show you smart. I told you that you can even do the same thing with smart.
Okay, okay.
There are tools like CDD search, PFAM, smart. Most of the time we like to use CDD search because in CDD search you can submit either your protein sequence or your nucleotide sequence. On the other hand, if you go with PFAM and smart, you need to use only protein sequence. PFAM and smart tools do not work with your nucleotide sequence. Is this clear? Is this
Yes.
clear?
Yes. Yes. Yes.
Everyone?
Yes, sir.
Fine. Now with the template sequence, when I submitted the template sequence to NCBACD research, it is showing this particular function, sodium hydrogen exchange. Can you see the domain function of this particular gene? This it is saying that this gene is involved in sodium exchange, right? Guys,
Yes. Yes.
yeah, say either yes or no. If no, I will explain again. If yes, we will move to the next step. If you don't say anything, I don't understand whether you are getting the things or not. Now take your GenScan output, okay? Take the sequence. You can take either nucleotide sequence or protein sequence. You can take any one of that one and check for the presence of the same domain. Check for the presence of same domain. I am submitting whatever I have mined from Grameen database with the help of GenScan. I am submitting it in the FASTA format. Okay, submit that one. Nothing has come here. Nothing has been displayed in the sequence. I mean the results page. So it means that what are the sequence that we have taken is not the same thing. Go back to GenScan page again. There you can see some more sequence below sequence, right? 780 bases. Can you see this one? Can you see this one?
Yes. Yes. Yes.
Yes,
Please
sir.
take that one and submit it in the NCBI CDD search. NCBI CDD search is not working. It's not working. Instead of NCBI CDD search, we will go with the PFAM or smart page. Take the help of other tools. Give me a minute. I'll get back to you.
Okay, sir.
Sir, I got the results in CDD search.
You got the result.
Yes, the same.
Same
Same
means,
which you did. Same result came.
I mean,
That result that you submitted the sequence from MCBI.
hmm.
That result, same result came, means
I mean, with the
sodium.
GenScan
Yeah.
output, with the GenScan output.
No, sir. This
No. What sequence you have submitted to 3D search?
uh,
GenScan output or NCBI output?
same with Gen scan output, and the result are same, which is for NCBI sequence and the Gen scan sequence. Both have the same result.
I'll do one thing.
This one. Yeah.
No, I'll do one thing. I'll stop sharing the screen. You please share the screen. You can share your screen now.
Yes, sir. I'm sharing.
Hassan, you can share your screen. Hassan, can you show us with the template sequence and then with this one, okay? Amazingly, I don't understand why it's not working in my thing and you got the answer, right answer.
So my
How about
screen is
others?
visible to you?
Please share
So
the
my
entire.
screen.
Yeah, we can see now.
Yes, sir. This is the result that I got, and I will show you the template sequence which I
Okay.
pasted. This one I have pasted. I copied from here.
Can you
This
copy
one.
again from, yeah, copy and show us.
Okay, sir.
2052, yeah, right. Yeah, please copy that one.
No, it's not showing, sir.
Sir, I think it's issue with the server, I think.
Actually, because we got 100% identical, right? It should display. It should display the result. Yesterday, I practiced with the same thing and I showed to my students in the class where I got the result.
Yes, sir, because of the server might be.
Maybe, but I mean, I need to show you the result, right? How it appears. Then only you can get the confidence that okay, whatever the sequence that we have taken, it is having the same domain, and whatever the gene that we have mined now, it is also having the same domain. Well, unless until it do not show the same domain, then we cannot call it the same gene family. Okay, is this clear? Is this clear?
Yes, sir.
We'll wait for some time. Please try in your systems also. I don't know why NC based EDD search is not working. Try again. Once you get, please let me know. The same sodium proton exchanger will appear in your GenScan output also.
Sir, sometimes CDD search won't work, sir. So
Yeah,
it
but
happens.
yeah, it worked for Hassan. Please check again.
It may be due to the internet and other things also. Otherwise, if we repeatedly use it also, sometimes it will not work. So it
That's
happens, sir.
true. But it happens, but we need to confirm, right?
Okay, sir. Okay, sir.
So should I stop sharing?
Okay, you can stop. If anyone get the result, you can share. Absolutely no issue. You can stop, Hassan. Fine. Guys, submit this again into CDD search. We'll see how it works. How about others? Only few are interacting with me. Rajarshi Mondal, Rakesh Tiwari,
Sir,
Suresh Gupta.
here also the problem with server, it is repeatedly means counting the time.
Okay.
One more question I am having, sir.
Yeah, please.
Sir, in case of this Arabidopsis mRNA, which I have taken from NCBI, in that particular mRNA, it is not started with the, it is started with the CCA or something, and it is ended with the some ACA.
Okay, if that is the
So
case,
it is giving the result. Actually, it gave earlier.
okay. The thing I got your question. I got your question. The thing is, sometimes after getting the sequence results, some people will trim the extra base space and submit the exactly the CDS portion.
Okay, sir.
And some people will submit along with the primers and everything. What are the extra sequence that they got?
Okay.
And if you look into that particular flat file, whatever the CDS that you have taken from Aeropsys,
yes,
if you
yes.
scroll down the flat file, there is an option by name CDS, right?
Okay.
The flat file, NCBI flat file.
Yeah.
You can share your screen because others
Yeah,
also
I'm
need to
sharing.
understand what you're trying to say.
Yeah.
You can share your screen.
Yeah, I'm sharing, sir.
Okay.
Visible to you, sir?
Yeah, it's visible. Please enlarge the NCBI page.
This one is
Yeah.
mine.
Yeah, I understand. I understand. First, you go back in the same page. Go back.
Back. Okay.
Back side. You have come to faster page, right? Yeah. Please scroll down. Scroll down. Vipul, scroll it down. Slowly. Yeah, come down. 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 Yeah, stop at the CDS place. Please stop at the CDS place. You see, don't click that one. Oh, okay, fine.
Okay.
Fine. The CDS is starting from 609. Can you see that one?
Okay. Yeah. Yeah, 609. Yes.
Yeah, it is starting at 609. If you click that one, now click the CDS. Yeah. Of all the sequence, your exact CDS sequence is starting somewhere around 609. There you can see ATG, it's there, right?
ATG, yeah, sir. Got it.
So, I mean, that's why NCBS is not a curated database. That's why we call NCBS as a non-curated database.
Okay, sir.
Whatever you dump in, it will be there. Very preliminary curation will be done, not the fine or the complete curation. Is this clear?
Yes, very much clear. Thank you very much.
Yeah, welcome. Guys, you again submit the same sequence to CDD search because I want to show how a gene characterization is done. For that, we need the answer. Submit that one. Vipul, you can stop sharing the screen. I will share my screen. Yeah, I'm sharing my screen, guys. Can you see my screen?
Yes, sir.
Yeah, fine. Now keep it aside the CDD search thing. Now in the next tab, open GSDS. GSDS. GenScan Gene Structure Display Server. Can you see this one? GSDS, Gene Structure Display Server. Can you see this one?
Yes, sir.
Guys, please speak out.
Yes, sir.
Or else I don't understand.
We are
Yes, sir, we are seeing it.
Yeah, please open that one. Open that one. Here you can see a bed format. Choose that one to FASTA format. so that you can see two boxes, right? One is CDS box input and the other one is genomics box, right?
Yes, sir.
Yeah. Now, in the CDS, you paste here 2502 sequence obtained in your GenScan. Listen carefully. In the CDS box, you submit or you paste your GenScan 2052 whatever you have got with the help of Grameen blast output, right? And in the second box, all the time faster format is mandatory. Without faster format, we cannot do anything. In the second box, submit your genome sequence, nothing but your sequence that you have obtained in your Grameen database, right? Or the blast output of your Grameen database, almost around 10,000 basis long sequence. Copy that one and paste it in the second box. Right? Is this clear?
Yes, sir.
Yes, sir.
In the first box, you paste the CDS sequence, whatever you obtained from your Grameen sequence. In the second box, paste the Grameen sequence, okay, that is your genome sequence. In the first box, you are submitting CDS sequence. In simple terms, from the around 10,000 odd bases, you got 2052 bases, right, in the GenScan. Now, when you submit CDS in the first box and genome in the second box, just click submit, don't do any other things. Submit and wait for a minute. Can you see the result on the screen?
Yes, sir.
All of you, can you see the result?
Yes, sir.
Sir, yes.
Fine.
On your screen, you're seeing this.
Fine. Guys, now see here. This is the first exon. This yellow color is represented by your exons, right? And this is the second exon, third exon, fourth exon, fifth exon, and this is your sixth exon. Whatever the sequence that you have taken now, okay, is having 1, 2, 3, 4, 5, and 6 exons. And the black lines, they represent your introns or non-coding sequences. And this blue color portion is nothing but your promoter sequence. Upstream to the start portion, so it is your promoter sequence. Is this clear?
Yes, sir.
Everyone. I know you all know splicing. Splicing is a mechanism where introns are removed, right? Exons will be brought together with the help of your lariat sequences, lariat and laviate. Once they are brought together, you will get a CDS. I am not talking about alternate splicing. I am just talking the basic splicing mechanism where from your pre-mRNA, what are the pre-mRNA that has been formed after transcription, contains both introns as well as exons. The introns will be removed by a mechanism called splicing so that you will get exons. And once you get the exons, that is called your mRNA. Later it will get into mature mRNA by adding 5' cap and 3' poly A tail so that it will become stable. Now, in today's hands-on session, you have taken a template sequence. From that template sequence, you started mining the same gene in another species, and today we have used sorghum species. You started mining that one or retrieving the sequence from that particular sorghum database. When you submit your sequence to the Grameen database, Grameen is much easier when compared to those things. That's why I use template sequence. You got a result. You open that particular result. After opening the result, you can see almost all the time, most of the time, the top hit would be your gene of interest that you are searching for. Open that particular sequence. By default, blast or the Grameen blast will take minimum basis. Go for the configuration of that particular page. Increase, generally keep 3000 upstream and 3000 downstream. Don't keep 10,000 and 10,000 upstream and downstream, because you may run into other gene. If there is no nearby gene, it's okay. But if there is some other gene, nearby gene, you will run into that gene. Don't do like that. Take 3000 and 3000. Now you check that one, remove the line numbers, again check that one. It will get automatically refreshed. You will get a much bigger sequence than the previous result. Copy that result and keep it in the GenScan, choosing Adopsis as the organism. Then what GenScan will do is, it will remove all the promoter sequences, it will remove all the terminator sequences, it will remove all the intron sequences, and it will give you the only exon sequences, which we are calling as coding sequences or CDS. Once you get the coding sequence, first all you need to do is you need to verify whether whatever the sequence, the coding sequence that you obtained is the same sequence that you are searching for or not. Why I am saying this is, all the genomes starting from E. coli to highly evolved human beings, it's only repetition of the 4 bases ATCG, 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 ATCG. But this ATCG pattern is making a dog as a dog, human being as a human being, cow as a cow. So we need to verify whether we are looking for the same gene or not. How we will do that one? Simply check for the presence of the domain that is there in a template sequence. This template sequence we have taken is about the function of sodium proton antiporter. Now we got that particular sodium proton exchanger in CDD search. The same domain, whatever that is there in a template sequence, should be there in a GenScan CDS sequence. Once both of them matches, it is a preliminary confirmation. It's a confirmation, not preliminary, it's a confirmation saying that whatever you have mined from the Grameen database, from the sorghum genome, is the same gene that you are looking for. Once you confirm that one, whatever the mined sequence is the right sequence, then move down to the next lane of the works, like GSDS. Nothing but you are characterizing the gene. Characterization in terms like that, you are searching how many exons or how many introns does it contain. Once you want to confirm, or once you want to confirm the presence of exon, number of exons, or the gene, you want to do gene characterization, simply open GSDS. Once you open GSDS, gene structure display server, you will get a bed format. Choose the FASTA format. Once you choose the FASTA format, you will get 2 boxes. In the upper box, submit your sequence in FASTA format. What are the type of the format that have been asked for, you submit in the same format. Don't submit in other formats, it won't recognize your sequence. And all the time remember, FASTA format means the greater than symbol should be there in the first line, and your sequence should not start immediately after the greater than symbol. You keep greater than symbol in the first line, and from the next line you start using your sequence, whether it may be CDS sequence or genome sequence, doesn't matter. A sequence to be called FASTA or FASTA alignment, which stands for fast alignment. The greater than symbol should be in the first line, and your sequence should start in the second line from the first place. Don't submit numerical numbers or any Latin, Greek like symbols because it won't recognize. It recognize only your four bases: ATCG, ATCG, ATCG. It won't recognize other things. Don't submit all the GenScan accession numbers or any asterisk symbols or your dollar symbols. Don't do those kind of things. It will be difficult for it to identify. Once you submit your sequence, the sequence will be displayed like what you are seeing on the screen. By looking at this particular sequence, you can say it contains these many number of exons and these many number of introns. See here, this particular intron is very, very big when compared to any other exon also. So most of the times, your intron sequence will be much, much bigger than your exon sequence. That way, out of 6 kb or 5 kb, whatever you have taken, it's showing only for 2052. Means the rest sequence contain your promoter sequence, if it is in the upstream. If you get any blue color after this 3 prime, it shows that you have even taken your downstream sequences. Right? Is this clear?
Yes, sir.
Yes, sir.
Guys, everyone, because if you're not clear, please let me know. I'm ready to explain again. If it is in physical interaction, that would have been much better, but we are in online session. Yeah, someone is asking something, please.
Yes, sir. So this last step, so GSGS. So you please explain it again, possible.
Okay. When you come to the GSDS, okay, I'll go back again. I'll show you the screen. Basically, when you open GSDS, it will be the bed format like this. You keep it to FASTA format. You will get 2 tabs, 2 boxes. Submit your CDS sequence, whatever you have mined using GenScan. Submit that one here. And the second one, you submit your genome sequence, whatever you have obtained in Grameen. Because it needs both introns and exon sequence. In the CDS, only exons are there. If you submit your genome sequence, it will contain both your introns and exons, so that it will show you in the form of exon and intron display. See here? Now you will get image like this.
So I'm asking about the rationale of this, means why we do this GSGS.
Can you ask the question louder? I'm unable to hear properly.
Sir, I'm asking about the main purpose behind this GSGS analysis.
Okay, behind this GSDS, without this GSDS, how can you come to know the number of exons and introns that are there in your sequence? Right? Are you able to understand my answer? Without doing GSDS, you don't understand how many exons are there or how many introns are there. You are characterizing the gene. Characterizing means you are trying to find out the number of exons or the number of introns that are there. And if you look at this image, it clearly says that the exon, the last intron, it is very, very bigger. Very, very bigger. Since you may get a doubt, I asked you to take 5,000 upstream or 4,000 upstream, whatever the sequence that I asked you to take, you have taken. Where that big length sequence is going waste? It is going waste in terms of your introns, in terms of your promoter sequence. Unless until you do GSDS, you cannot identify the number of exons that are there in your CDS. Is this clear?
Yes, sir. Now clear.
Okay. Any other doubts?
So you have not shown the results of SMART and PFAM for validation.
For SMART, you need to use the protein. You can use the same SMART and PFAM also. Take the SMART or PFAM and submit protein sequences, not the nucleotide sequences. Why we use CDD such frequently? Because why do you prefer that one is you can submit either your nucleotide or protein sequence. If you go with PFAM, Smart P, some other tools are also there, and other protein based. You need to submit only protein, right? You can exercise that one, and what are the doubts you are having? Please post in the workshop WhatsApp group. I'll get back to you. And whoever wants to work on this kind of works, please choose your gene of interest so that we can have a meeting, a private meeting. If one or two are there, we can have a private meeting and we can work on that one. But before going,
Sir, already I have taken one. We will work.
okay, madam, before going that, please read the paper that I shared in the chat box in
Sir,
the start
actually,
of the class.
meanwhile, I got disconnected, sir. So, if I get it again, or somebody else can
I
share
will
it.
share that one again. Yeah, already Dheeraj has shared that one. He's one of my student. I'm sharing the paper again.
Yes, sir. I got it.
Did you got the paper?
Yeah.
Yeah. Please open that paper, which was published in last September. It's about genome-wide analysis, not associations. I'm talking about analysis only. And Suresh Gopi has asked a question at 7:54. Can we do it for one more gene? Yeah, Suresh. What I suggest is, please, I think most of you are working on a particular topic. You choose that particular gene. If you're working on human systems, blast against your thing on human database. If you're working on a plant systems, choose your plant, what exactly you want to do. And in this paper, I had written very clearly, everything is crystal clear in that paper. What we have done, how we have done. I had provided all the links, detail, web servers, tools, databases, whatever I had used there, I had mentioned in detail in that paper. Please go through that paper, you will get every information. And wherever you have any doubt, please ping me in the WhatsApp group. I'll get back to you at the earliest. Or else you can reach me through my mail ID. I'm sharing my mail ID again in the group in this chat box. Whoever want to get in touch with me or who are interested in learning these kind of things, want to put in the paper, please get back to me through my email ID. Once you mail me, please introduce a one-line introduction is enough. I'll get back to you. Uh, I get back very quickly through emails. Okay.
Fine, sir. We will work.
Yeah, maybe it will be nice if you share in WhatsApp group only so that others can also learn. That would be good. That way, you can paste the screenshot. That's the idea of collaborative working. So that there might be same questions which Anil need not repeat it if he answers once in WhatsApp group. Okay.
Any doubts?
Sir, actually, this is very exciting lecture today. So actually, I'm using these tools many times. Means most of the times I'm using it regularly. But the thing is, the way you explained is totally different. The approach, where you started and where you ended is totally different.
Okay. If anyone is interested in carrying out any kind of this kind of works, please let me know. We can continue it as a project and we can publish it paper. From Bioclues, more than 52, 55 papers have been published from this kind of workshops only. And we are starting from founder to everyone. Everyone is a good listener. They're ready to work with any of you. We are open to work with any of you.
Already, I do have one paper with the Bioclues women team. And
that's super great to
I
hear.
like the way of this group working. So I like to do more with the Bioclues team.
Yeah, nice to hear from you about Bioclues and the paper. Others, if you have any
Sir,
quick question, yeah, Hassan,
yes,
please.
sir. So can we do the same analysis with the fungi as well?
Can
Fungus?
we do the same analysis in?
Fungi.
Yeah, first, before doing the in silico work, you first check whether the genome has been sequenced or not. You can do in any system. You can do in any system. The tools will be
But in
varying little bit. The
fungus,
tools will be varying little bit.
sir, just 1 minute, sir. In fungus, the gene expression pattern is different. It is not like plants. So when you are doing this GSDS, it may not work properly.
No, no. If the genome is available, we can check with the GSDS also. Not exactly the same GSDS. That's why I'm saying the tools will be little bit varying, but the core concept will be the same. Right?
Okay, sir. Thanks. Thank you, sir.
First, check whether the sequence is available or not. Then start working on that one. Read some 10 to 20 papers, 10 to 10 good papers, peer-reviewed papers, so that you will get much information, and then start working on that one. And whenever you encounter any things, you please let me know. I'll get back to you. Already I've given my mail ID also in the. It's Anil Kumar OU not zero. Sorry for the typo. It's Anil Kumar OU @gmail.com. Right? Please save this. I'll get ping me anytime. I'll get back to you the next day, mostly the next day, or whenever I see the email. I have a question of the topic. I need to get the list of the plant pathogenic bacteria. Where can I look for it? That's what Rakesh I am saying. First, you check for most of the bacterial pathogenic bacteria are also sequenced these days. You first check whether the genome has been sequenced or not. If it is sequenced, then you can play with the genome.
Sir, he is asking only the list of pathogen that he can get from any crop pathology book. He can
Any gene, any disease, whatever you type, you'll get some genes responsible, like blast, trots, everything you can open.
No, sir, he is asking for only pathogen name.
Blast disease, which you would call by Pyricularia oryza. Okay. I'll send that list to you, Rakesh. Please send me the mail. Okay. Am I audible to you? Okay, fine.
Yes,
No,
sir.
you please send me a mail. I will send a reply to your mail. Okay.
Sir, again, in this last tool, it is
one by one, please, people. Yeah.
Yes, sir. So when we were using the last tool, means in which it shows that your file is too big or something. So again, we have to be very precise when we are pasting something. It's
Last file
showing
means which file you're talking about?
this one, gene structure display server.
Okay. This one?
GSDS.
Yeah, this one only, right?
Yes. Yeah.
Where did you face the problem?
Means, yeah, it continuously showing that there are so many lines of errors coming when I'm using the same.
Did you submit first of all it in the FASTA format?
Yes, it is in FASTA format.
See, I'll submit again in front of you only.
No, sir. No worry.
Just I'm hearing your doubts. Okay?
Yes.
The second tab, I'm pasting the genome sequence. Sometimes, if there are any gaps there between the sequences, it will be submitted as different sequences. So it will give error for. Sometimes the servers may not work properly. You please be patient and try it again 2 to 3 times.
Okay.
Sometimes there is a problem with servers also. I do accept. It may not be manual. You will get the result.
Yes.
Right?
Yes. Wonderful.
Fine.
Thank you.
Some more is asking doubt. Who is he? Any other doubts? Guys, if you want to ask me, please ask me. Generally, if you're not getting any doubts, means there are two concepts mostly. One is you have understood everything, and second thing is you have not understood anything. If you understood something, obviously you should get doubts. Which I need to go with, that you have understood everything or you have not understood anything.
We understood few things, sir. I will get back to you when we
Yeah,
post up in
I
mail.
know, Dheeraj, you know these kind of things because already you had a publication in this particular area. How about others?
So yeah,
Yes, sir.
whatever you explained, we understood. Now we need to go through the paper and
Mhm.
then plan further. Yes, after this workshop, maybe we can get back on this topic and go into details for
Anytime you can
publication.
get back
Yeah.
me through the email, I'll get back to you. If there are no doubts, can I call it for the day? Because I have some other meeting at 8:45.
Sure, sir.
If
Thank
you
you, sir.
have any
Yeah,
quick
I think
questions, please let me know.
yeah, I think we should thank Anil for his gracious time and wonderful talk. Thank you so much.
No, Prashant. Actually, I am very thankful to you. You had given me an opportunity to share my knowledge.
No, no, no. At all.
I'm
Last time,
thankful to you.
we remember in 2021, Anil had comparative genomics. That was something also on the sidelines of sodium antipotar, proton antipoters work. That his lab has excellently done on in sorghum bicolor. Okay, yeah, that was a wonderful thing. I still remember. So all of you, please switch on your camera for a wonderful photograph with Anil. Anil, there are two wonderful, other two wonderful plant biologists, Yasin and of course Pranita. I think, they'll be very happy, to collaborate with you. You all could write and contribute, one wonderful paper, actually. These two are also excellent scientists. Yeah. So, yeah. Yeah. And
Thank you.
we should
Nice.
also thank, Rajul, for recording today. So much, Rajul, for your wonderful
Thank you very much, Rajat.
recording. Yeah. Must be pretty late for that. Yeah.
Actually, pleasure is mine in interacting with young, experienced everyone, so that I can learn something from you and you can learn something
Yeah,
from me.
what do you call, one thing. If no one is probably switching on your camera, then you're a bot. This is very clear from now. Yeah.
Yeah.
Peter Abu, Rajeshri, Manav, Pooja Salumke.
Other, please switch on your camera so that we can have a
Pooja, I
photograph and
requested your address, please. Please, would you mind Pooja sending the receipt? We usually send the receipt like this. As soon as you register as a life member, we send you 3 emails. There was no response. So kindly send your full address for posting the address. Okay.
Yes, sure, sir.
Suresh, Gopi, Vijayanta, Deepika, Zion. Yeah. Yes, thank you so much. I'm taking a photograph. 1 minute. I copy paste. Sorry, 1 minute. 1 minute. 1 minute. Yeah. Yeah. So say cheese. Yes. Thank you. Thank you so much.
yeah. Thank you all. Bye.
Thank you.
Thank